Hello, my name is Joyce and I will be presenting about experiment 9.1, precipitation of protein by organic solvents, ethanol and acetone, and experiment 9.2, measuring protein concentration using Lowry protein assay. There are four parts to this presentation. First is the introduction, which I will discuss about the pre-lab knowledge, explaining concepts and principles, the objective of the experiment and its importance. Next, I will explain the process, followed by the results and interpretation of the graphs and figures obtained. And finally, I will give my reflections regarding this experiment and the whole class in general. For experiment 9.1, we did protein precipitation, which is the separation of protein from extra contaminants. This is important in protein precipitation and is usually done by adding organic solvents such as acetone, ethanol, methanol, or TCA. However, in this experiment, we will only compare the efficiency of acetone precipitation versus ethanol precipitation, in which proteins are insoluble in both organic compounds, causing the impurities to dissolve and leave only the proteins. For experiment 9.2, Lowry protein assay was used to determine the protein concentration. This made use of copper-2 ions in alkaline conditions, reducing it to copper-1 ions, and was reacted with Folin CuSl2 reagent, causing the solutions to form a blue color in which the amount of protein is proportional. These experiments have one main objective each, which was to compare the efficiency of acetone and ethanol precipitation and to measure protein concentration using Lowry protein assay. These experiments are important because it demonstrates how the protein was purified using ethanol and acetone precipitation, and it determined which one is more efficient than the other. Also, it gives an understanding of the principles beyond Lowry protein assay, a type of protein concentration determination method which could be useful in protein studies. Here is the step-by-step process of the experiment. But before proceeding to the experimentation, it is important to note that safety is always a priority in every lab, so be sure to wear lab coats, eye protection goggles or glasses, and gloves as seen in the picture. For the precipitation process, the samples were prepared accordingly and were exposed to centrifugation, air drying, and resuspension, while for the Lowry protein assay process, 8 test tubes were prepared according to the table shown. Appropriate reagents were added and exposed to incubation each time a reagent was added, and the absorbances of each test tubes at 660 nanometers were determined. Moving on to the results and interpretation. This table shows the absorbance at 660 nanometers with Folin CuSl2 reagents with different dilution of BSA solution. As you can see, the higher the BSA concentration in mg per ml, the higher the absorbance value. Next, the calibration curve was plotted as shown in the slides in order to get the equation of the trend line, which is to be used to calculate the protein concentration in mg per ml as shown on the next slide. Looking at the table and the equations, we obtain the protein concentration through ethanol and acetone precipitation, which were 0.16 mg per ml and 0.56 mg per ml respectively. Based on the results obtained, it can be concluded that acetone precipitation is more efficient than ethanol precipitation since it yielded a higher protein concentration value. As for my reflections, in this experiment, I learned that I learned about the two methods of protein precipitation, which are ethanol and acetone. both of which are organic solvents that proteins are insoluble to, and also the process of protein concentration determination through Lowry protein assay, in which I was able to come up with the conclusion that acetone precipitation is a more efficient method since it yielded a higher protein concentration than ethanol precipitation. For this class, due to COVID, it was divided into online sessions and on-campus sessions. For the online sessions, I learned how to utilize apps such as ColorName to help in the identification of the pKa of each solutions. I also enjoyed using Pymol and visualizing proteins in different colors. Lastly, I think the most useful website I learned from the online session is the NCBI protein database, which I am still using in my senior project right now. But of course, I find the online session very underwhelming since we weren't able to be in an actual chemistry lab using actual materials. So I kind of didn't enjoy it that much. Hence the meme shown on the slide. Then we move on to the on campus sessions. I was really satisfied with the experience. At first, I struggled with manual pipetting, as shown by the meme on the slide. But now I could do it more efficiently and faster too. I also enjoy the changing of colors of the solution and using spectrophotometer to measure the absorbances. It was enjoyable but tiring since we have to do pre-lab, lab notebook, and post-lab, which is why sometimes I make silly mistakes such as using an upside down gel in my lab reports. But overall, I find the pre-lab, lab notebook, and post-lab useful since I still look back on it until now as references. That is all for my presentation. Thank you.